So, good day. I'm Menandro, David Jr., and I will be reporting about Benedict's test, urine glucose. So, let's start. So, what is Benedict's test? So, Benedict's test is a chemical test that can be used to check for the presence of reducing sugars in a given analyte. This test can thus be used to identify simple carbohydrates that include a free ketone or aldehyde functional group. The test is based on a compound combination of sodium citrate, sodium carbonate, and the pentahydrate of copper, sulfate, known as Benedict's reagent, also referred to as Benedict's solution. So this is the Benedict's test. So when exposed to reducing sugars, the reaction undergone by Benedict's reagent result in the formation of a brick red precipitate, which indicates a positive Benedict's test. So an image detailing the changes in the color of the Benedict's reagent from clear blue to brick red that are triggered by exposure to reducing sugars. So this is the picture. So Benedict's test principle. So a reducing sugar becomes an enediol when heated while being in the presence of an alkali, which is a relatively powerful reducing agent. Benedict's reagents, cupric ions, are converted to cuprous ions when reducing sugars are present in the analyte. These cuprous ions combine with the reaction mixture to produce copper oxide, which precipitates as a brick red substance. So this is the picture. So this is an aldose, Benedict's reagent, carboxylate ion, and the brick red precipitate. So Benedict's test procedure. This is the preparation of Benedict's reagent. So Benedict's reagent may be created by combining distilled water, 100 grams of sodium carbonate, and 173 grams of sodium citrate with 17.3 grams of copper sulfate pentahydrate. So this is a required quantity. So here, the sodium carbonate works as an alkaline medium. The copper sulfate serves as the source of Cu2 ions, and the sodium citrate interacts with the Cu2 positive ions to form complexes. Distilled water is used as a solvent. So next, testing for reducing sugar. So 1 milliliter of the analyte sample must be mixed with 2 milliliters of Benedict's reagent and heated in a bath of boiling water for 3 to 5 minutes. So the development of a brick red colored precipitate of cuprous oxide confirms the presence of reducing sugars in the analyte. So this is it interprets the results of Benedict's test. So color of the precipitate and the gram percent of reducing sugar. So green, we have 0.5%, yellow 1%, orange 1.5%, and red 2%. So what does Benedict's test detect? So the main use of Benedict's test is to find out if an unknown analyte contains simple carbohydrates. The presence of reducing sugars with free aldehyde or ketone functional groups can be detected with this assay. Either a monosaccharide or a disaccharide can be the reducing sugar. So what is Benedict's reagent? So Benedict's reagent, often referred to as Benedict's solution, is a complicated chemical compound made composed of a sodium citrate, sodium carbonate, and copper sulfate pentahydrate. So, Benedict's reagent turns brick red when exposed to reducing sugars and other reducing agents, changing from clear blue to red. So how can Benedict's test be performed? So 1 milliliter of the analyte solution and 2 milliliters of Benedict's reagent should be combined in a test tube to perform Benedict's test. This combination must next be cooked for 3 minutes or so in a hot water bath or until a visible change in color occurs. So, how is Benedict's solution prepared? So, by combining citric acid and molecules with cupric ions from copper sulfate pentahydrate in a basic environment given by sodium carbonate, Benedict's solution or Benedict's reagent can be created. One can use the finished product to detect the presence of reducing sugars. So, this is some substances that give positive results from Benedict's test for Benedict's test. Some example of substances that yield positive results for Benedict's test are listed below. So, first we have glucose, second fructose, and third ribose. So, that would be all. Thank you for listening.